Okay. Good morning, everyone, and welcome to the Executive Roundtable session here at Investing in Cures Summit 2021, presented by the Foundation Fighting Blindness and the RD Fund. My name is Jason Menzo, and I am the Chief Operating Officer with the Foundation Fighting Blindness and also a member of the management team with the RD Fund. In addition to the ASL interpreter that's on the screen right now, we also have closed captioning available for today's session. You can turn on the captioning by selecting the CC icon in the bottom right-hand corner of the video window. Additionally, you may ask questions during today's session. Please submit your questions through the chat or the Q&A modules that are located to the right of the video player on the screen. All features are available by tabbing through and then selecting by pressing Enter on your keyboard. I'd now like to introduce my co-moderator and good friend, Dr. Rusty Kelley.
Greetings from Raleigh, North Carolina. I'm Rusty Kelley, Senior Vice President of Investments and Alliances at the Foundation. I joined the Foundation approximately 3 years ago to help our CEO, Ben Yerkes, and the management team and our board launch and lead the Foundation's venture arm, the Retinal Degeneration Fund, or the RD Fund. The RD Fund's sole member is the Foundation, and our investment decisions are governed by a stellar and diverse board of directors, independent of the Foundation, led by Chairman Warren Thaler. If the audience would like to learn more about the RD Fund, please visit our webpage, rdfund.org. My colleague, Jason Menzo, and I are excited to introduce our panel of trailblazer executives to this audience. Jason and I will introduce each speaker, followed by a brief presentation of their respective companies. These executives are all pursuing genetic approaches to curing disease and/or preventing disease progression. We have a preclinically staged company in SparingVision, rapidly approaching the clinic. We have 3 clinically staged companies in Atsena, Iveric, and Gemini. Iveric and Gemini are publicly traded, and Gemini has taken advantage of the recent rise in SPAC mergers to enter the public markets. And I'll shamelessly note that Atsena and SparingVision were both co-founded by the RD Fund. Let's start the discussion by first introducing Mr. Patrick Ritschel, CEO of Atsena Therapeutics. Pat Ritschel is a seasoned startup executive and entrepreneur. Prior to Atsena, Patrick was a co-founder and president of Stride Bio, a gene therapy company based in Durham, North Carolina, and continues to serve on that board of directors. Prior to Stride, he was an early angel investor and played an instrumental role in the formation of gene therapy company Bamboo Therapeutics, acquired by Pfizer in 2016. He spent his early career with Monsanto in St. Louis and Hong Kong, and he's an active in the rare disease community and currently serves on the board of directors of the Friedreich's Ataxia Research Alliance. Pat, thanks for joining this panel. The floor is yours.
All right. Thanks, Rusty. Appreciate the kind words and introduction. Really happy to be able to be here today and to talk a little bit about Atsena and what we're doing. So Atsena Therapeutics, as Rusty mentioned, was incubated and started just about a year ago with investment from the Foundation Fighting Blindness through their RD Fund, and they participated as well in our Series A investment that we closed last year. Atsena is an ocular gene therapy company. That's who we are, and that's what we do. And we're founded really based on the technologies that came out of our co-founders, Shannon and Sanford Boys lab at the University of Florida. As Rusty mentioned, we do have a clinical program, which we were happy to bring in, the LCA program, and I'll talk a little bit about that as we go forward. We also have several preclinical programs that we're working on and advancing towards the clinic, and I'll talk specifically about the Ushers program on today's talk. Beyond that, we have some interesting technologies as well, some dual vector technology that helps support and drive forward the Usher program, and some AAV capsid technologies, both for intravitreal and subretinal administration. And so I think we've done, put together a program here, a company that's got a foundation based on some interesting AAV technologies, along with preclinical therapeutics and a clinical stage asset. And over the course of the year, we've really moved things pretty rapidly forward. We believe in the power of genetic medicine to help inherited retinal diseases. We are focused on IRDs specifically. and we're very patient focused as well. I've been in the rare disease community now for quite a few years, and our founders and of course Foundation Fighting Blindness all keep the patient focus directly in front of us. And our goal is really to move our programs through the preclinical and clinical development so that ultimately we get them to patients. I'll talk a little bit on our clinical stage asset, the LCA1 program. So this is a program that originated out of Shannon and Sanford's labs probably more than 15 years ago. And we were happy to be able to bring that into the company in July of last year. And so we're in the middle of a phase 1/2 trial. This is a single site trial at the University of Pennsylvania with Dr. Jacobson as the PI. And we're currently enrolling. We've gone through the first 2 cohorts in our dose escalation phase, and we're in the process of moving forward, and we'll look to enroll the 3rd cohort this summer. Once we've gone through the dose escalation phase, we'll be moving forward to choose a dose and go into an expansion cohort, both in adults and then opening it up to pediatrics. So we're excited to advance this program. And I'd like to mention that we do have a paper that came out just about a week ago from our PI, Dr. Jacobson, that talks about the early results from the first cohort. So I'd encourage folks to take a look at that. And I'll just say that we're pleased with the early results, good safety and encouraging signs of engagement from the therapeutic. We like this disease for a couple of reasons, not the least of which is that the retina is very preserved. So we're able to deliver the gene to structure that's intact, and by delivering a functional copy of the gene to intact retina, we believe we should have high odds of therapeutic success. Our preclinical program that we're going to talk about today is Usher's 1B. And so USH1B is a disease that affects not only vision, but also hearing and vestibular function. We're focused on the vision piece. It's a bit challenging from a gene therapy perspective because it is a large gene. And in order to, from a gene therapy perspective, there's limitations to what can be packaged inside an AAV capsid, and this gene is too big to do that conventionally. So we're using a dual vector technology that, in layman's terms, splits the gene into 2 pieces, packages each one independently, and then as they're delivered to the cell, they'll recombine into a functional full-length cDNA copy and thus work effectively. So this is a little bit complicated technology, but we've done a lot of work on this, and the early results look very promising. We are preclinical stage, but we're working forward step by step and look to bring that to the clinic as rapidly as we can. We also have a third that's undisclosed preclinical program, and we'll probably be making an announcement on that in the second half of this year, with the goal of having really 3 programs in the clinic within the next 18 months or so. And I guess I'd like to talk a little bit about the technology piece too. The dual vector technology enables us to go after diseases like Usher's, as well as potentially future indications that also have large cDNA. The capsid technology that we've in-licensed originated out of our founders' labs. I think we've got something that's pretty game-changing from a subretinal standpoint. So subretinal is a surgical approach to delivering. And one of the keys to having success in gene therapy is ability to deliver to cells that you want to target. We have some capsids that really spread well within the eye and are able to cover a larger portion of the retina than conventional or natural serotypes do. And so we think this is pretty game-changing, and we'll be utilizing that as we go forward with our preclinical programs. We do recognize that intravitreal administration is an easier route, and that's something that we're spending some effort on to develop capsids that meet the needs of the therapeutic indications that would be appropriate. We've got some capsids we've brought in already that we think look very, very attractive, and we're going to continue to engineer on top of those to develop, really potent and robust capsids for future delivery. So, in a nutshell, what we've put together here is a company that's based on solid technology. Like, we're taking very appropriate and proven steps to deliver to targets and indications where we have a high degree of confidence of success. And I think we're very pleased with the support that we've gotten from our early investors, such as the Foundation Fighting Blindness, as well as our current Series A investors, and looking forward to moving our programs forward. So happy to take questions or address any other comments as we go forward from here. Rusty?
All right. Actually, this is Jason. Thank you so much, Pat. And we will hold questions till the end. We've got 20 minutes at the end of the broadcast, or at the end of the session, dedicated exclusively to a Q&A session. So it's actually my pleasure now to introduce our next speaker, Stéphane Boissel, who is the CEO of SparingVision. Stéphane was actually appointed as the CEO of Sparing concurrent to the first closing of the Series A financing for the company in August of 2020. He was previously the chairman of the board of the company. And prior to joining SparingVision as the CEO, Stéphane earned his, actually served as Executive Vice President of Corporate Strategy for Sangamo Therapeutics, based in San Francisco. And he joined them back in 2018 on the back of the acquisition of TxCell SA, where he was the CEO of the company. Stéphane is an experienced biotech professional who brings over 25 years of leadership across corporate finance, strategy, and business development, along with executive leadership. It's my pleasure to introduce Stéphane.
Thank you, Jason. My name is Stéphane Boissel. I'm indeed the CEO of SparingVision, and I'm joining you today from Europe. So SparingVision was started in 2016, and as Jason just said, we closed the second part of our Series A in August 2020 with fund coming from different US and European investors, including the RD fund. SparingVision is a genomic medicine company in ophthalmology. Ophthalmology is the only thing we do and the only thing we will be doing. And genomic medicines are the only tools or technologies we will be using. Our lead asset, SPVN06, is a gene therapy that is mutation agnostic. And I will explain to you what does that mean. It's an AAV-based product that encodes for a cargo that is a combination of a neuroprotective or neurotrophic factor, and an enzyme. And the combination will lead, as the objective to lead to the preservation of cones at a stage of the evolution of the retinitis pigmentosa disease, where patients have lost all, if not most of their rods. And the idea of this cargo is a science that dates something like 20 years, when the scientific founders of the company realized that despite most mutation leading to RP affecting the rods primarily, at one point in the evolution of disease, the cones were also dying. And so over the last 20 years, they have identified and characterized the mechanism of action of this neurotrophic factor associated with an enzyme. The neurotrophic factor, which we call RDCVF, which is short for rod-derived cone viability factor, is working at favoring the uptake of glucose into the cones. And this is a protein that is normally secreted by the rods. This neurotrophic factor is associated
Great. This is Rusty Kelley. I believe we have a slight interruption. So let's move on to our next speaker, and we'll come back to Stéphane. So our next speaker is Dr. Walter Straps. Walter is the Chief Scientific Officer of Gemini Therapeutics. And prior to Gemini, Dr. Straps was most recently Vice President, Discovery Biology at Intellia Therapeutics, where he led internal and external CRISPR-Cas9 therapeutic programs through discovery into IND enabling studies across a range of tissues and indications. And prior to Intellia, he served as director RNAi therapeutics at Merck & Co. and helped Merck integrate and build up its RNAi therapeutics group after its acquisition of Cerna Therapeutics in 2006. And prior to the Cerna acquisition, he was a scientist at Cerna. Walter, thanks for joining this panel. The floor is yours.
Thanks, Rusty. I'm going to give you a little bit of an idea of what Gemini is all about and the programs that we've been working on, similar to my colleagues have done. So Gemini is a clinical stage precision medicine company focused on the development of new therapies through a deeper understanding of disease. Our initial development focus is on a heterogeneous disease. It's known as dry age-related macular degeneration, or dry AMD. And we're applying our insights in genetics and biology to identify patients that carry genetic variants that place them in the high-risk category, and then treating those patients with a medicine that is designed specifically for the deficiency that they have. Our lead product candidate is known as GEM103. And as I say, we're going to treat patients who have dry AMD. So for those of you who may not be as familiar with AMD, it's a progressive, irreversible disorder of the macula. AMD generally has been categorized as either wet or dry. The overwhelming majority of the patients actually have dry AMD. So of the 16 million patients with AMD in general, about 90% of those patients, about 15 million of them, have dry AMD. Over time, dry AMD causes what's known as geographic atrophy, or GA, so I may use that abbreviation, and that occurs when regions of the macula are replaced by scar tissue. The resulting blurry vision, loss of night vision, and loss of central vision as the disease advances affects about 1 to 2 million people. And also currently there are no approved therapeutic options for these patients. Some of you may be familiar with wet AMD, where there actually is a treatment. So the anti-VEGFs, the VEGF inhibitors, and so on. For dry AMD, there's no such treatment at this point in time. So these patients essentially become progressively, lose their vision progressively. And because vision obviously is central to independent living, it has a large and growing societal impact, especially as the population ages. So our lead product candidate, GEM103, it's in phase 2 development for the treatment of dry AMD, specifically in patients who have a loss of function mutation in the CFH gene. GEM103 is literally a recombinant version of that protein. So in those patients, we believe it will address the complement hyperactivity, the hyperactivity of a particular pathway within the eye, and overall dysregulation of that complement pathway, resulting in improved retinal health in these particular patients. So we've got a couple of imperatives that guide the work that we're doing at Gemini. First, we aim to replace the complement inhibition, what we'll call orthodoxy, using an approach that leverages the better understanding of the pathway and the genetic predispositions of the specific patient population that we are treating. So we believe that this is a differentiated approach that will address complement dysfunction and maintain retinal tissue homeostasis. So second, we also intend, as I've sort of alluded to redefine AMD as a disease of genetic subtypes, so that essentially we're going into patients who have a particular genetic variant or genetic defect and treating those patients specifically with a treatment that should be most beneficial to them. In dry AMD, we believe that genetic analysis implicates the variants in the gene that codes for a protein called complement factor H, or CFH, is representing that largest subset of patients. We believe that the precision medicine approach is optimal to address this burden and to restore regulation of the complement system. And we can use genetic assays, biomarkers of the pathway that we're looking at, and natural history studies to match the therapies to those patients who have the underlying genetic defect. So many studies have demonstrated the role that genetics plays in the development of AMD in general and dry AMD specifically. We have a current natural history study, which we refer to as Clarity. And what we're looking at there is essentially a patient population that carries specific genetic defects and seeing how the disease, the dry AMD, progresses in those patients. The goal there is to make our subsequent interventional trials, our later clinical studies, more powered for those particular class of patients. So by understanding the progression of the disease in those patients, we can better understand how to treat those patients. We are currently developing GEM103. It's in a phase 2A trial that we have a name for. It's called Regatta. The trial completed its enrollment in February, and it is expected to have an initial readout later this quarter. In those patients, we're going into those patients that actually, again, carry mutations specifically in the complement factor H gene. So this augmentation of complement factor H or CFH activity represents a unique approach to address the imbalances in the immune system. Essentially, within these patients who have dry AMD, the immune system is running sort of out of control, and our goal is essentially to bring it back down and under regulation. So we are confident in the substantial opportunity that exists to help patients with GA. We see further potential for patients with a non-genetic population that may benefit from GEM103 as well. We look forward to exploring that space. I won't go into a huge amount of detail, but we also have AAV gene therapy programs targeting the same pathway in patients who are earlier in the phase of disease. So patients who may not have started to see retinal damage yet, but based on their genetics, we expect that they will. I guess we'll get to questions a little bit later. Thank you.
Right. Well, if anyone at home was wondering if this is live, I think that we've got the proof of the pudding. This is the way that it goes. I actually thought for sure it was going to be either Rusty or my Beagle that was going to be the proof that it was live, but it's the way it goes sometimes. So very excited. We have Stéphane back with us, and so I'm going to turn it back over to Stéphane to Stéphane, why don't you just quickly recap the first couple minutes and then pick up around where you left off this at the stage.
Yeah, sure. Sorry for that. That indeed is the inconvenience of being live. I don't know whether I was cut off, so I will just kind of summarize SPVN06, our lead asset, which is the first mutation agnostic gene therapy to be developed in RP, retinitis pigmentosa. This gene therapy is an AAV-based product that has two transgenes. One is a neurotrophic factor that we call RDCVF, rod cone-derived cone viability factor, and the other one is an enzyme, and the two transgenes are two isoforms of the same gene that was discovered and encoded by the scientific founders of the company something like 20 years ago. And they came with this idea about when they realized that most mutation in the RP disease were affecting the rods. And despite that fact, at one point in the evolution of the disease, the cones were dying. And so over the last 20 years, they have identified and deciphered the mechanism of action of this combination of a neurotrophic factor and the enzyme, which are two subproducts, so to speak, of the same gene that they encoded back then. This product aims at preserving what is left of the vision upon diagnosis. So it's not a typical gene therapy in the sense that we are not aiming at restoring a vision that is lost, but the idea is to go at a time when most patients are diagnosed with RP, i.e. at a stage where they have lost their rods, and so they have lost let's say their night vision on some of or all of their peripheral vision. Then they come to the physician and then they are diagnosed. And at that time, gene replacement therapy targeting the rods is not possible since the rods are gone. So the idea would be to inject SPVN06 at the time of diagnostic and to preserve what is left of the vision, to preserve the cones which at this stage are still pretty much working, and which, as you know, are critical for the vision. So the idea would be to make sure that those patients upon diagnostic will be able to have their vision preserved through the rest of their life and not have the clock ticking, unfortunately, which is the case today as there is no treatment at this stage of diagnostic of the disease. We aim at starting our first in man with this product later in 2021. We are in the final stage of putting together the IND and CTA package. We aim at conducting this first in man trial both in the US and in Europe. That will be a typical dose escalation study with 3 different doses on 5 different cohorts. And if everything goes well, we should transition that product into what could potentially be a pivotal study sometimes in 2024. Next to SPVN-06, our lead asset, we are expanding the portfolio. I was expecting to be able to announce today our first acquisition, but you will have to wait for a few days. That will be announced, if everything goes well, next week. And this acquisition will give us a second product, which is another mutation-agnostic gene therapy product. But unlike SPVN-06, which is working on the metabolism of the cone, this second product, which we will call SPVN-20, will act on the function of the cones. And the idea would be to reactivate, so to speak, the function in cones which have lost their ability to function as part of the evolution of the disease. These are called dormant cones, which you find in the late stage of the evolution of the RP disease. Both SPVN-20, this new asset, and SPVN-06 will be developed first in retinitis pigmentosa, but the idea is to explore those assets in other rod cone dystrophies as well as in cone dystrophies and possibly other degenerative ocular disease. Next to that, we are expanding the portfolio through various business combinations. The aim is to create an unprecedented portfolio of genomic medicine in the space, both mutation-agnostic gene therapy, gene engineering products such as gene regulation or genome editing-based product, but as well as reprogramming tools as we see a future, probably in the very long term, in turning some cells in the eye into critical cells for the vision, such as cone. But this is for the very long term. The immediate future for the company, again, is to go in man with its lead product, and this should be achieved if everything goes well as soon as 2021. This is it for me. Thank you very much for your time, and I'm turning over the floor back to Rusty, I guess.
No worries. Very good. Thank you so much, Stéphane. And just a quick reminder before we introduce our last speaker that if you have any questions, or if you'd like to participate in the Q&A, which we'll have about 20 minutes of Q&A after Glenn speaks here in just a minute. But that Q&A interface is in the page on the right-hand side. You can chat any questions that you have in right there. Our final speaker for today is Glenn Sblendorio, who is the Chief Executive Officer and President of Iveric Bio. He joined the company in 2016 as Executive Vice President, Chief Operating Officer, and Chief Financial Officer, and then was appointed to President in January of 2017. Prior to joining Iveric Bio, he served as the President and CFO and a member of the Board of Directors of the Medicines Company from 2006 to 2016. And Glenn also served as the Executive Vice President and Chief Financial Officer of iTech Pharmaceuticals from 2002 through 2005. He currently serves on the board of directors for Amicus Therapeutics and Intercept Pharmaceuticals, and we're really excited to have him here today. So I'll turn it over to you, Glenn.
Well, thank you, Jason, and thank you to the Foundation for inviting us today to speak at Investing in Cures. And I've been fascinated as I listen today to my other colleagues talk about the efforts to find cures through gene therapy or treatments for patients in diseases where there's, in many cases, no treatment. So it's really exciting to hear what they had to say as well. So the focus at Iveric Bio and our goal is to develop transformative therapies for retinal diseases. So we're a company that is completely dedicated to finding either treatments, therapeutics, or gene therapy solutions for these devastating diseases. And I'll talk about our programs in a moment. I will mention, since I think we may be the only public company here, I may mention some forward-looking statements and refer you to our documents filed with the SEC for a full discussion of risk and uncertainties. Our public company trades on NASDAQ under the ticker ICSEE. And I want to talk in more detail about our very diversified portfolio. As I mentioned, therapeutics, we have 3 programs, largely focused on major market opportunities, AMD type opportunities, And as you heard before, there's a number of efforts, and there are some existing treatments for wet AMD. I'm going to talk about ours in a moment. And the second goal of the company is to speak is around gene therapy. And these are for inherited retinal diseases or orphan diseases. But in order to bring these programs through, we have assembled an outstanding team of people with a lot of experience here. Of course, you need good science and medicine to develop drugs or gene therapies for these, but people are very important. And we have a board of directors that has extensive experience in ophthalmology, led by Dr. Adrian Graves, who's an expert in research and commercialization here. Dr. Mark Blumenkrantz, well known at Stanford. Dr. Cal Roberts, CEO of the Lighthouse Guild, and also Dr. David Geyer, retinal specialist, publisher of many articles, and quite a KOL in the field of retinal medicine. Our team is also very experienced, and it's strategies led by Dr. Dougal, Praveen Dougal, in this business for over 27 years. And importantly, on clinical operations, it's led by Evelyn Harrison, who's done over 25 trials in retinal medicine and involved with over 10 NDAs. So you need great people to move these programs forward. Let me start talking first about our therapeutics. We talked about complement today. We also have a complement inhibitor, a C5 inhibitor called Zimura, for the treatment of geographic atrophy secondary to dry AMD. And I think you heard some news before about a program that Walter has. Well, ours targets the bottom of the cascade. There is, as Walter mentioned, there is no treatment for geographic atrophy, and it affects over 5 million people worldwide. We have completed one phase 3 trial, which we call GATHER1. It was completed in October of 2019, hit its primary endpoint, and showed a reduction in the growth of GA of over 27%, and importantly, a very favorable safety profile. Our second trial, which we started last year in July, after a pause because of the pandemic, the COVID pandemic, started in July, recruiting well. We expect to finish the second trial in the third quarter of this year. And if we hit our primary endpoint at the 12-month point, we intend to file an NDA for treatment of GA secondary to dry. So a very large market, no treatment opportunities here. I will mention that during this pandemic, we've worked very hard to recruit patients. We put in a number of procedures, not only at the investigator level, but also for the patients, to keep our patients safe. And I think that's why we are on track to finish our recruitment for the second trial in the third quarter of this year. So looking forward to that. Our second therapeutic trial, also using Zimura, or C5 inhibitor, is for Stargardt disease. This is the most common inherited macular dystrophy in both children and adults. This affects approximately 62 to 77,000 people in the US and Europe, so quite a large orphan disease. We have a phase 2B trial currently recruiting, and we expect to recruit up to 120 patients. Again, this is a disease with no treatment, so look forward to the results there. Our third therapeutic, much earlier in development, which we call IC500, it's an anti-HTRA1 inhibitor with potential application against geographic atrophy and potentially other AMD. So this is a relatively new target. There is a large clinical trial ongoing here. It may be yet another tool in our toolbox to help patients in the devastating diseases of macular degeneration. Let me move on now to our gene therapy programs, and we have 5 of them. We have 2 lead programs. Our first called IC100, which is for RHO, autosomal dominant retinitis pigmentosa. This is a disease that impacts approximately 11,000 patients in the US and in Europe. And we're hoping to see this gene therapy in the clinic in the latter part of this year. We're happy to be working in collaboration with both the University of Pennsylvania and the University of Florida on this program. Our second gene therapy program, called IC200, is for BEST1 related diseases, again working with University of Pennsylvania and University of Florida on this. We would also like to see this in the clinic potentially at the end of this year as well. Best disease, the prevalence of that disease is somewhere between 10 and 40,000 patients in the US and Europe. We then have 3 programs. We're working with the University of Massachusetts, and I think we heard a lot about large genes and their ability to treat retinal diseases. We have 3 programs with the University of Massachusetts using the mini-gene technology. And that is essentially taking a large functional gene, reducing in its size so it fits in existing AAV vectors and retaining the functionality so that when the gene is delivered, it is functional. And we have 3 programs there. Our lead program is for LCA10, the CEP290 mutation, smaller orphan disease with 3,000 to 4,000 people in the US and Europe, but obviously a very devastating disease. And here, this is in preclinical development, and we're looking to identify a preclinical candidate later in this year. Our second program, which we hope we can offer patients both a solution in Stargardt disease therapeutically, but also with gene therapy. This program is targeted at ABCA4, which is Stargardt disease. Some very interesting preclinical work there, making good progress. We intend to have an update there later in this year, but a very, very important program for us. And then finally, our third mini-gene program is for USH2A. This is a disease that impacts about 20 to 60,000 patients in the US and Europe. And this is also in early stage research, but good progress there, and hope to report some good news on this program as we proceed. So each of these devastating orphan diseases, a variety of solutions, some using full-length gene therapy, and in the case of the work at UMass, a mini-gene concept. So 8 products in our program. We're very happy to hopefully moving forward and offer some of the treatments and solutions to patients. So I'll finish by saying that our number one priority, since it's late stage, large indication, is to finish GATHER2, our GA trial. And as I mentioned before, we're on target to finish recruitment in the 3rd quarter, and then hopefully we'll look for positive results as it reads out. So Jason, thank you for letting me round out the speaker session.
Excellent. So I'll kick off our Q&A session with a lead question, and then we'll move through the panel of speakers with a slew of questions from both Jason and the audience for that matter. So the lead question. So with genetic approaches to precision medicine, we usually understand well the target and its mechanism. Therefore, the primary challenge is a pharmacological challenge, and that's selecting an appropriate ocular delivery strategy, achieving safe and efficacious expression levels of a gene in the right patient cohort, the appropriate cell type in the retina, to intervene at the right time, and to select the appropriate endpoints or measures of safety and efficacy. Natural history can help inform these product development decisions. And I should say that of all the resources the foundation brings to bear, I've learned to appreciate most its comprehensive natural history studies, a significant resource to all stakeholders in this field, including to support industry. For the broader audience, patient natural history is the relationship between genotype, visual function, and disease progression, and the selection of the appropriate functional and structural ocular endpoints, including the use of patient-reported outcomes. Can each of our speakers discuss their approach and/or the challenges to understanding patient natural history? And perhaps we can start with Walter first, given Gemini's significant investment in the Clarity natural history studies that includes custom genetic testing that screens for high-risk genetic variants associated with dry AMD in the complement system.
Sure, thank you. So yeah, it's critically important, the natural history piece of this. So in the case of dry AMD, for example, there's a well-established link between the onset of dry AMD and carrying particular variants in complement factor H. What is less well understood and is really the underlying thesis that we want to test within our Clarity natural history study is the progression of that disease, and whether or not the rate at which the disease progresses is driven by genotype. So onset for sure, but is the rate at which the disease progresses also driven by those genotypes? And if so, are there particular genotypes where it is more aggressive, the disease progresses faster, or less aggressive, and so on? With the goal, again, there being, as we look to interventional trials, which are the patients that we think would benefit most cleanly from our treatment, or most quickly begin to show a therapeutic effect from that particular treatment.
Great. How about Glenn?
So, Rusty, in many of the diseases that we're all collectively working on, natural history obviously plays an important role, but these are diseases with unmet need. So in the case, as Walter was just talking about in GA, they're targeting upstream, we're targeting downstream, and we're learning a lot about this disease as to, how we slow the progression and where the right place in the complement pathway may be to block. And this is not, this is more about, what's going to be best for these patients. We know that the AMD roadway that starts with early stage drusen and ends in geographic atrophy or death of the eye is only now starting to be understood. Although we have a detour in that roadway as it progresses with wet AMD, there is a treatment, but it does put you back on that roadway towards GA finally. So we're learning a lot, I think, through good science and good clinical studies. We're building our database here, but still a lot to be learned because most of the diseases we spoke about today are unmet medical need with not a lot of development or clinical experience.
Thanks, Glenn. Thanks, Walter. Pat, what do you think?
Sure. So I do think natural history is a really valuable tool in terms of understanding the disease and understanding the variability from patient to patient and the progression over time. One of the challenges that we have, we work in IRDs specifically, which is our smaller patient populations and generally not as well studied, and so it is a more challenging environment. I think one of the biggest challenges that you have in rare disease across the board is numbers and longitudinal depth, and so the earlier a natural history study is started, the more patients that participate in it, and involvement of foundations to help those things happen, and centers of excellence, really make a difference to drug development in the long run. But it's an early investment, and it takes time to bear fruit. A short natural history study is often not as informative as what you'd like, and getting years of natural history with large N number enrolled is important.
That's great, Pat. Great advice. Start early. And so, Stéphane, you're obviously racing towards the clinic. Is this something that you're investing a lot of time and resources understanding?
Yeah, absolutely. I mean, natural history studies has from the start been very high in the agenda of SparingVision. We are conducting 2 studies, one retrospective on 110 patients in Paris, and one prospective. The retrospective is over, and we expect to report at the upcoming ARVO the main findings of that study, but they will speak to the correlation between structural and functional endpoint, and we've learned a lot from that study. And the prospective study, which is ongoing, halfway through the recruitment of something like 80 patients, will be used as a leading study for the not the first in man, but what could come next as possibly the pivotal study, and will also, of course, inform the main clinical endpoint that we will be looking at. But again, this is obviously something which is critically or totally fundamental before embarking into a clinical study to understand the population you are going to treat, but also to make sure that you have the right set of endpoint in your clinical trial.
Okay, thanks, Stéphane. Jason?
All right. Thank you, Rusty. Actually, I wanted to go. We have a couple questions that have been chatted in the chat feature, and I'll pose it to the group, and whoever feels like tackling it first can. The first is something that I know you've all thought about, but I don't believe have talked a lot about, at least not to my knowledge. And that's about drug delivery. And so with ophthalmic indications, particularly IRDs, retinal indications, the current standard of care is fairly invasive, as you all know. And so curious what you all think about the future of drug delivery for these inherited retinal diseases or dry AMD. And if no one jumps in, I'll pick someone. How about that?
I'll jump in maybe just quickly. I think, clearly, yes. Some of these therapeutics are delivered by surgery, others by intravitreal injection. It is invasive. I think that everybody, I'll be a little presumptuous, but I think everybody in the field really wants to move to deliveries that are as easy and less invasive as possible. It's better for the patient. It's better from a safety perspective all across the board. So I think there's definitely an effort to do things as simply and as safely as possible. Having said that, I think there's still a fair bit of work to be done to get to that point, and I don't see that changing in the near term.
Anyone else have any thoughts on that they want to add to the conversation?
I think I would agree with that. I mean, ultimately, what it boils down to is you need to get the drug to the place that it needs to be, and that in most cases, frankly, that means you've got to be fairly invasive. You've got to go, into the vitreous or subretinally in some of those cases. So it's something aspirational, but at this point, the science basically dictates that you need to be invasive to get the drugs where they need to go.
Yeah, I mean, if I may add on something, Jason. I mean, today nothing has proven to be better at transducing the cones or the rods than the subretinal way. But we all expect that in the future, hopefully not so long in the future, we will have access to different route of administration. And this will be of particular importance if we want to reach out to, highly prevalent disease.
Yeah.
So, Jason, I'll agree with my colleagues, and I'll just mention, and I think also in the larger indications that are still using therapeutics and hopefully gene therapy in the future, we're all looking for extended delivery, especially in the therapeutics, because even intravitreal injection, although it's well tolerated, we'd like to see longer duration. So, looking at solutions, there are many things in the works, but I think we need a little bit more time to be a little bit more efficient and take away the patient burden.
Understood. Let me pose another question to the group that was chatted in, and that is around the impact of COVID. And this is one of the few times I think that, regardless of whether you're in Europe or the Far East or in the US, really anywhere across the globe, we're all facing equal, while different, but equal challenging circumstances with regard to COVID. I'm curious how each of you think COVID will have an impact, not just on your short-term plans, but maybe your mid and long-term plans with regards to patient care or clinical trial enrollment. How are you thinking about the impact of COVID on your business?
Jason, maybe I can start having a clinical trial ongoing in the COVID world. We were ready to start our second phase 3 trial in February of 2020 and made a very difficult decision, which turned out to be the right decision, but hindsight, that we should pause because it was an uncertain world. What was going to happen to the investigator sites, what was going to happen to patients. Would you start a trial that would have a lot of patients missing visits? And more importantly, could we potentially put the healthcare providers, the investigators, their staff, and patients at risk? So we took a pause, got beat up pretty bad by Wall Street because that created some uncertainty, but to be expected. And thought a lot about that and worked with our sites, worked with a number of different groups and put in a program that kept the patient as safe as we possibly could. What was that? We provided car service. We provided single drivers. We provided PPE equipment, not only to the patients, but to the sites. We ensured that as we brought a site up, that there was adequate protection for the patients. A lot of interaction between the company and the sites, and the patients to be sure that they felt a safe environment. So as we started this second trial in GA, we not only are focused on recruitment, but on retention as well. So there's a lot that goes into bringing a patient on board and keeping that patient safe during the trial. It is a new world. So where priority may have been in the past recruitment, retention now is just as important, if not more important. So that's the way we've dealt with it. Implications for the future, I think it changes the way we do things in the future, but more to come on that. But it's an emerging situation that changes every day.
Pat, how about you guys? How are you thinking about the impact of COVID?
Yeah, so I think I agree with a lot of what Glenn had just said. I think that from our perspective, we had a similar, a smaller situation, or a single trial and a smaller number of patients. But I think the issues are the same, and going forward, there's a lot of work that people are doing in terms of what can be done remotely versus what can be done on site, and I think those conversations will continue, but. There are certain tests that you have to be physically present for, and you can't necessarily do remotely. And so I'm not sure exactly where this ends up. I do think that there's going to be a move towards more remote type things, but I think that's also a shift over to longer term, and it's got to be technology enabled. There's certain things you just can't do at your home. But there are, maybe things that people will look at in terms of bringing tests to patients rather than bringing patients to centers. I think there's definitely, work that's gone on during COVID to look at things like that. So.
Very good. And I'd be remiss without getting a European perspective. So, Stéphane, any additions to how you guys are thinking about the impact of COVID?
Well, in all fairness, we've not been really impacted, probably because we were not yet in clinical development. And so we had a few weeks of delay here and there, but it's fair to say that we have not been impacted. And I mean, looking at the future, I'm not a COVID specialist, but it looks like the disease should be under control soon with I guess, vaccination. And so we can just expect that, we won't be as impacted. I mean, the industry won't be as impacted in the future as it has been in 2020. But it's again, time will tell. But I'm more optimistic with the availability of the vaccine on the way COVID specialists are projecting the disease to become like a chronic disease, but in some way under control.
Yeah. Okay. So let me do this. We're going to have Rusty make some closing remarks in just a minute. I've got one final, we're going to call this the rapid fire section. One final question I want to pose to each of our speakers. And if you could answer it in just a few words or in a sentence or less, but really want to capture what you're most excited about over the next 2 to 3 years in regards to the inherited retinal disease and retinal degeneration space. So Walter, we didn't get your opinion or your perspective on COVID, so we'll go to you first for the rapid fire, and then we'll work our way around to the other speakers.
Sure, thanks. I think the thing for us that's most exciting at Gemini is that we're going to be going into those later stage trials, and from the scientist perspective, which is what I am, to test that hypothesis around whether or not, in the case of dry AMD caused by complement factor H, whether or not supplementing complement factor H in those patients does what we hope and expect it will do, which is slow the rate of GA progression.
Awesome. Pat, one sentence. What are you most excited about?
I think we're most excited about seeing readouts from our ongoing clinical trial and moving our 2 programs into the clinic as rapidly as possible with novel capsids that we think are going to be, I think, really game-changing from a subretinal perspective.
Awesome. Stéphane?
Yeah, well, obviously we expect to be well, it looks like my signal is poor again. Sorry for that. We are expecting to be starting our first in-man trial soon, and this is obviously a very exciting milestone for us.
Very good. And then finally, Glenn.
So very excited to see our 2 lead gene therapy programs hopefully get in the clinic by the end of the year. No treatment for patients. And of course, I have to mention our GA trial. Excited to finish that and hope for good results so that, again, we could provide a solution to all those patients with GA.
Excellent. Well, thank you very much. I'm going to turn it over to Dr. Rusty Kelley for some final remarks here to close out the session.
Great. Thank you all for joining today's session. Thanks to Pat, Walter, Stéphane, and Glenn for participating. We very much admire you and your companies, and we look forward to supporting you in any way possible, whether that's via the foundation and or the RD Fund. Thanks to our partners, Iveric Bio, Regenex Bio, Biogen, and Charles River. Please join us for our next session in a few minutes as Ben Yerxa and Jean Dewan, CEO of the foundation and the RD Fund, and Jean Dewan, a board member of the RD Fund, lead a discussion with an impressive list of ecosystem panelists. To join our next session, please close out this session, go to the session schedule, select Join Session to join our next session. And thanks again for joining Investing in Cure Summit. Take care and goodbye.